Thank you for having the opportunity to talk to you as a neuroscientist, as a neurobiologist. We are all defined even by name, by the name neuron. So everybody expects that the brain is just running by neurons, and all information in computation are done by neurons. And what it turned out is, and that will be my talk will be about, is it's even more complicated. There are even more cells that play a role. Not that it is not already enough to have neurons. This is just an approximation to have like 86 billion Milliarden Neurone. You have lots of capillaries. The brain only weighs 2% of our body mass, but it uses 15% of our oxygen. So in terms of energy consumption, it's not one of the organs that saves us energy. We have here, it's in miles, we have 100,000 miles of axons in the brain. If you put them one after another, also that, of course, all approximations. Even if I said that in terms of the body, it uses a lot of energy, think about a computer that just uses 25 watts of power per day, and still could be an artificial, an AI machine. So you see, this is what the brain has to deal with. We have lots of other stuff involved here. We have 10 trillion synapses that do the computational work of neurons. So neurons get input from approximately 10,000 other neurons at the same time, and have to process them and have to convert them into action potentials and into a code that then will be transmitted to other neurons. And to be honest, we are still trying to discover what this code is. For some motor neuron areas, it seems to be clear now. So one can now, just by thinking about lifting an arm, lift a robot arm. But in all other areas of the brain, it's a lot more complicated, and we still don't know what the code is. As I've said, so far, our focus has been on neuronal connections and also of connectomics. That's a new area, neuronal connectivity, that tries to map the brain in terms of all the axons connected to other neurons, what's basically the map inside the brain, how connections are made. Keep in mind, 100,000 miles of axons need to be connected. We are far off, even using artificial intelligence, even using thousands of scientists looking at it. We can map like 200,000 neurons and their connections. Keep in mind, there are 86 billion we have to map in the end. So we just a tiny fraction. So if you look inside this picture, so probably what we can do is even smaller than the size of this red arrow here. It's probably the white arrow inside the red one that we can map. The rest is still open territory. But you can see imaging gets a lot better every day. So the connectivity of the brain is on the way of at least on a certain scale to be discovered. You see here, these are neurons made visible, in this case of the visual cortex. You see here neurons and their axons made visible. Keep in mind, this is just like 12 or 15 out of 86 billion. You see here how it is. You can also do some computation, and you can do you can look how neurons are connected to each other. There are excitatory connections and there are inhibitory connections. And one secret of the human brain is actually that it has more inhibitory connections than excitatory connection. Looks like German bureaucracy. But what it determines is that the inhibitory connections are important to get certain rhythms into the brain and to determine how is the flow of information. So that's why inhibitory neurons are very important components. And we have a lot more inhibitory synapses than any other species, even more than chimpanzees in proportion. But you see what the task is. I mean, find the flow of information in this 10 micrometer cube of neuronal tissue. So that's a very, very complicated task in order to find out what is the code, what needs to be changed. And also, once you have done it, basically this cube of neuronal connections in one moment in time is not the same as the next moment in time. Whenever we experience something, whenever we learn something, the hard drive of the human brain is actually changing, because we do have a component that is called structural plasticity, which means that not only the weight of connection is changing, but also the direction and how information is processed. And also for synapses, at least some computational work shows that a synapse is not just on or off, it has 26 grades of shade, so which means shades of gray. So it has at least 26 different states of strength and not just 0 and 1. That's another thing that makes things more complicated. But so far, what I've told you is a synaptic connection from one neuron to another. But as you can already see in this graph here, we have other components which are usually ignored. This is a glial cell. It's called an astrocyte. And we have approximately the same number of glial cells in our brain as neurons. And still it's called neuroscience. And people have so far more or less ignored these glial cells because, I mean, the translation would be glue. There's a glue in the brain, they stick together, they help neurons for energy support. But now what becomes apparent, they are also involved in shaping the brain, and they are also involved in computational processes. So this is already, I think, complicated enough, but now you have to take other players into account as well. You also have to take into account modulatory inputs of other neurotransmitter systems which predetermine the synapse, if it is actually possible to change it or not. Some of them are involved also in motivation, stress, concentration, and so on. What also became apparent is that also the immune system actually plays a role. We have one type of glial cells in the brain, probably 10% of all cells in the brain belong to the immune system. They are microglial cells, which are also in the title of my talk. And it took us many years in my group, since we worked with these cells, to convince the neuroscience community that they play an important role also in the interaction of what happens in our body, and convert this information into the brain in terms of immune response. So the brain is responding to the immune system, and also the brain is influencing the immune system. And people didn't believe that. It was a kind of a dogma until, I would say, 2010, 2014, that whatever happens in the body does not happen in the brain, and there's no counterbalance. Why? Because there's a blood-brain barrier that inhibits any immune signaling into the brain. Which is kind of funny, because at the very beginning of my discipline of memory research, the immune system was instrumental, because in the 1960s, people discovered that antibodies in our body actually have a kind of a memory of a surface of a bacteria or a virus, because the B immune cells, they are called B memory cells, they actually produce antibodies that are specific to a receptor or a molecule on the surface of pathogens, and will then fight these pathogen if it invades us again. So if we survive it, it comes a second time, we can fight it. That's also the principle of vaccination. And it was discovered that antibodies work like that, which also in a way means, in a conceptual sense, these antibodies do have a memory of some surface components of virus, bacteria, or fungi that has invaded us before. Neuroscientists at that time had no clue how memories are stored in our brain. And they thought, well, but probably there are proteins as well. That would be kind of cool. Like the genetic code we have just heard before, we would have a code of proteins for certain memories. If that would be true, then we could design an experiment, and people have actually done that. If you train rats to avoid the dark, which you can if they always get a heavy and painful electric shock. Why is that important? If you give a choice to a rat to be in the open or in the dark, it will immediately go into the dark. It's a natural instinct to avoid predators from above. If they get a painful electric shock, after a while, they ignore the dark side and stay in the light. In this case, in these experiments that actually have been done, the rats were then immediately killed after they have learned it, and their brains were fed to other brains. And at that time, actually, the naive rats learned faster. So the scientists at that time took that as a proof that there must be some storage, and even it was discovered later, or it was thought it was discovered, it's not true in the end, that scotophobine is a protein that codes for avoiding the dark. That's why the name came on. At that time, it was very rare that a neuroscientific discovery would made it onto the front page of the New York Times. But in this case, actually, it made the front page of the New York Times, because if that would be true, you wouldn't have to listen to this talk. You can basically just eat me or eat my brain. So the New York Times came up with the headline, "Eat Your Professor," or you have a startup giving certain pills for certain languages before your next vacation in Italy or France or Turkey or wherever. It turned out not to be true. What they have just fed to other rats was a stress hormone, and proteins do not survive anything that do not survive the stomach, so they get put into pieces, except actually for prion proteins. So the experiment could not be repeated. It's not true, but it shows you there is an original thing between the immune system and the nervous system. And microglial cells are the connections. They scan all the time, like Terminator does, here the environment. They scan immediately, is the homeostasis interrupted or not? There's lots of surface molecules that do that. I spare you the rest. Look, if there's something in the brain happening, it could be an invasion. I mean, it's an immune cell, but actually the microglial cells do a lot more. They also shape neuronal systems. They help with the myelination of axons, which determines the speed. They actually look at synaptic connections, if there are some during development which are inactive for a long time, so belong to neurons that are not connected, they are deleted. So microglial cells do eat also dead cells. They fight viruses that enter the brain, but in particular, they are also involved in shaping the brain. There are even some molecular signaling which from astrocytes might actually signal to a microglial cell or to a neuron that certain synaptic connections should be deleted because they come from a neuron that's injured, that's invaded by a virus, or it's a neuron that's basically misplaced, which means it doesn't get any input and also gives no output to other cells. Which means the microglial cells do have processes. Here, this is a microglial cell. And if they see certain surface components, molecules on the surface of synapses, they actually eat that synapse away, which can make processing of information a lot more efficient, because all the ones that you don't need are gone, and you can go more directly with less computational power to all the places you would like to go. It can remove neurons which are inefficient. It can remove injured neurons, but it's also a real problem for the aging brain, because this is part of what happens during Alzheimer's disease, that the microglial cells are active all the time and they start to eat away synapses that we actually need. Which means for your conceptual view, keep in mind, we do have a presynapse that you are used and all know from school and have seen a post-synapse. But in addition, we do have at the synapse astrocyte end feet, which play an important role for the removal of neurotransmitters, in this case of the excitatory neurotransmitter glutamate. And we do have microglia processes, which all the time scan, is this synapse still necessary or not? And some of the synapses you have learned from your French classes in school, you might have lost during the years because you haven't used them for many years, and responsible are the microglial cells who have eaten them away. As I've said, they all the time scan their environment. This is here a microglia culture from our lab. And you can see here, they're always fiddling around and try to see if everything is in perfect order. This is also movies that we made, where you see that the processes of microglial cells touch axons in this case, and have a look if they do have surface molecules, which determines that they should be deleted. And here again, you see how from an electron microscopic picture, synapses are surrounded by microglial cells and by astrocytes. And what's important is that for information processes, microglial cells might actually promote the appearance of synapses. They might what we call pruning. They might eat away synapses, a term that we kind of borrowed from the botany people. And they might actually even influence if new neurons can be born and can be functionally integrated. And if microglial cells are activated in a pathological way, you can get diseases. For example, if they do not control where synapses appear and where they should go, people can actually, if that happens during development, can develop autism spectrum disorder, which is a disorder that's partly due to the fact that synapses that should have been deleted have not been deleted. And many people from the autism spectrum disorder report that very often they are overwhelmed by the sensory inputs. That's why they also like to do repetitive behaviors in terms to avoid this overflow of information of all the synapses that should have been removed by microglial cells, which are misfunctioning. In addition, microglial cells also do release a substance that's promoting the growth of synapses. It's a brain-derived neurotrophic factor, also something we analyze in our lab for many years now. It's also a factor that is known to promote memory processes. It promotes synaptic and neuronal plasticity, which is important, again, for learning and memory processes. And it also secures the survival of neurons. And one of the effects in the aging brain and also in people, in the brains of people who have Alzheimer's disease, is that BDNF is missing. And part of that at least what we try to discover is it the BDNF released by neurons, or is it the BDNF released by microglial cells? Because also microglial cells do age. They are also involved in what we call sickness behavior. So when we have a flu, we kind of usually start to stay away from people. We go to bed, we are grumpy, and we try to be socially isolated, which is a behavior. And this behavior is determined by the immune system and by microglial cells, which then translates this information to neurons, to certain parts in our brain. One is the insular part of the cerebral cortex, which kinds of gives us pleasure in being alone all of a while. Evolutionary, it makes sense because we cannot make other people ill from all the viruses and bacteria that we might spread by sneezing. So actually, in that sense, it could have been known for a long time from the sickness syndrome or sickness behavior that we are prone to change here. So the microglial cells are important for neuronal processes, for synaptic rearrangements, but they are also important for inflammatory processes in our brain. And that's a connection to when this process is not working properly, we get to disease like Alzheimer's disease. And here you see all the genes that are involved in neurodegenerative diseases. And you see for all of the diseases, from Huntington frontotemporal to Alzheimer's disease, you see an activation of microglial cells as well. So it could be that Alzheimer's disease is also not just the loss of neurons of synapses, it can be caused by microglial cells, which are also in this disease in the middle of the processes that are that are possibly important here, and they are activators of these processes. Some of them are maladaptive. For example, people with obesity have a lot higher risk of getting Alzheimer's disease. They have also neuroinflammation going on constantly. Also, sepsis is a risk factor. Also reduced physical activity. So it's good that you have a break time after the talk and the panel discussion to walk around a little, because this is what keeps your brain going and not stops it from aging, but the aging of the brain is reduced if you activate your brain all the time. And also what you avoid is neuroinflammation going on, which means microglia cells being active all the time and doing that. Let me finish my talk with a quote, how important our memory processes are for us as humans. You have to begin to lose your memory, if only in bits and pieces, to realize that memory is what makes our lives. Life without memory is no life at all. Our memory is our coherence, our reason, our feeling, even our action. Without it, we are nothing, is the claim here. So without our memory, we lose the connection to our world. And Alzheimer's disease is one of the diseases where we lose our memory, and it's at least that's what we believed, partly triggered by a maladaptation of microglia cells. And one of the early signs of Alzheimer's disease is the loss of synapses, and they all get eaten up by microglia cells. And the question is this a response to something that's going on earlier, or are the microglia cells actually the ones that are responsible for the loss of synapses? That there might be actually involved as a causative agent is suggestive by the fact that there's something going on that's called inflammation, that in all our body we have a lot more inflammatory sites when we age, but also in the brain we have a lot more inflammatory sites. So it could really be that the microglia cells are the driving force of the disease, why it's so important to study them. So in terms of microglia, the good, the bad, and the ugly, we need them for the development of our brain. They support synaptic growth. They are very important for the selection of synapses, what's called synaptic pruning. And synaptic pruning is not something bad, it's something we need for the efficient transport of information. In the adult brain, we need it for learning processes, but also we need it indeed to fight an invasion of viruses, bacteria into the brain, to activate the immune system to fight them. Aged microglia cells might be the ugly side, actually, because a dysfunctional microglia cell can actually do real harm to the brain and could at least be an accelerator of diseases, probably even the cause of it. With that, and with the people who did the work in my lab concerning the role of microglia cells for development, for the adult brain, for learning and processes, and probably also as something that does harm to the brain, are shown here. Thank you for your attention. Thank you very much.
Thank you very much for a nice talk. Any questions?
Yes, thank you for your talk and for humoring us. So I found your theory or the explanation of autism spectrum disorder quite interesting. And from what I know from a layman's perspective is that several other neurological development disorders, like, I don't like to call them disorders actually, like ADHD, look very similar in the brain. So is there a similar explanation for these, especially ADHD?
For ADHD, the explanation is, in this case, it's also the wrong selection of synapses, but in this case is that the power of certain subtypes in the brain that use modulatory neurotransmitters like dopamine and noradrenergic, they are not developed enough. Sometimes, and you need them in terms of to give priority to certain informations, so to certain inputs, and ignore others. So these are the modulatory neurotransmitters. And the synapses, either in numbers, are not enough, or the power of the synapses is not large enough because there's not enough dopamine release or not enough receptors. And then you have less power and you are more easily distracted than in let's call it a normal development. So in that case, it's partly due to the fact that synapses are not developed in a way it is supposed to be, but there it's a loss of synapses. There are other, let's say, syndromes where there's also too much synapses. For example, a genetic disease is a Fragile X syndrome. And also there, it's actually that the competition between synaptic synapses is not working. So also these children have too many synapses that are too weak. So there it's very similar to the autism spectrum disorder. But for ADH, for the ADHS, it's a different component. It's a development of certain subsystems that is either delayed, then as an adult, it's grown out. But what is always ignored is there are many adults which are also still do have ADHS.
Thank you.
Thank you. Any other questions?
Martin, when you started, you showed this complexity of the brain, including the millions of miles and so on and so on. How far away is brain science internationally from understanding the brain in total?
If we compare to physics, we are a long time before Planck. So, and if you compare that to physics, which means we are probably not 100 years behind because methods are increasing. But what is, I mean, there's a conceptual difference between physics and biology. In biology, we always come with solutions and then ask for the question. In physics, you first have the questions and the theory, and then and that's different in our case. I don't know for good reasons or not, but that makes it very difficult to find the right answers if you don't have a theory how it could work, because then it's hard to distinguish what is the right answer, just to go to the very complex questions of all. How does our brain get consciousness? I mean, how does that evolve? Why is it even necessary? You will not find two neuroscientists which will agree on the same definition of consciousness. That's something we have in common with philosophers. And then if you don't have a theory, it's very hard to design an experiment that will then fulfill your hypothesis or not. So that's one problem. Another problem is indeed, I mean, not making fun of it, it's indeed an extremely complicated endeavor. Because, I mean, you have, as you said, so many billions of neurons. You have 100,000 miles of axonal connections. And in addition, what I try to convey in my talk is that's not even enough. We have even more cells that are not just bystanders. They are actually shaping the brain as well. I mean, it's always hard to predict the future, as you know, but my impression is it will take us a long time before we can rebuild the system.
Okay, so thank you again for the nice talk and discussion.